afternoon. Good afternoon, comrades. Today, it's a great pleasure to have Ilya Nemenman, and I'll give a brief introduction. So Ilya started his physics career a long time ago in Belarusian State University, from which he transferred as an excellent student to Santa Clara University in San Francisco, where he got his bachelor's degree in physics, which he followed by a master's degree in physics in San Francisco State University, after which he went to a graduate school in physics in Princeton University, where he was working with Ilya, thinking about information theory in biological systems. And that line of thought occupied him basically for the rest of his career, among other things. And after his PhD in Princeton, he moved as a postdoctoral fellow to the University of California in Santa Barbara, and then to Columbia University, where he was working on applications of information theory to practical problems in inference in various biological systems, including nervous systems and also genetic circuitry in cells. After which, for a number of years, was a staff member in Los Alamos National Laboratory, this time in the, I forgot which group?
Computational science.
In the computational science division. And since 2010, he has been professor of both physics and biology at Emory University in Atlanta. He did many interesting, important things, both in theory and very applied research with biologists and biophysicists, experimental biophysicists. His work has been recognized by he's an American Physical Society Fellow and the Simons Investigator. And today he's going to share with us his recent thoughts about how to think simply about complex biological systems.
Thank you, Anton. Thank you. Well, thank you for having me here, and also thank you for making time and coming here to the talk. I'll try to not disappoint. The talk always runs long, so I'm going to start with the last slide, the important slide. This is the people actually who have done all the work that I'm going to be showing here today. Audrey Soderberg is now a faculty in Minnesota, and Mia is a graduate student. She was an undergrad. Much of this work is her undergraduate honors thesis. She's now a graduate student in New York, and Vijay is a faculty in North Carolina. And then there are various collaborators who helped our group along the way. So before I start, actually go to the main part of the talk, I just want to spend maybe a minute on what it is that we do and give you a few snippets of various different things that my group has worked on. And then maybe at the end of the lecture, if we have some time to talk with students, maybe you'll ask questions about those, right? So my main interests, as Anton has said, are about how biology processes information and learns from that. And that can be on scales all the way from molecular circuits. We adapt to the statistics of the outside world, to nervous systems, of course, and things like evolution, which is learning the structure, the statistical structure of the environment and trying to adapt to it, right? And so along the way, we worked on systems like, for example, mammary gland formation in mammals, inflammation. How does the system, our inflammatory system decides whether to inflame or not? This is a work done with Wilru, where we try to understand how the worms learn from the outside environment and make various different decisions. And even working with experimental friends with birds, where you put headphones on a bird and make a bird learn songs that they normally would not learn, and try to understand how exactly and how quickly they do those things. And so we have work on all of this. If you want to ask questions afterwards, I'm happy to elaborate on all of those things. What I'm going to start talking about today is that, I work in biology, birds, worms, inflammation, et cetera, but I am unapologetically a physicist. And so the question that maybe the first thing that one should ask is, why should the physicist study life, right? Why should the physicist study life, and brains in particular? Most of my work, most of the talk is going to be about neuroscience. And so here I go back to my sort of 13-year-old version of myself when I read this beautiful phrase by Immanuel Kant, that there's only two things in the world that feel my mind with unimaginable awe, and this is the infinite depths of the starry skies above my head and the infinite depths of the moral thought inside my head. You know, ever since 13, I pretty much believe that these are like two most important things to study. How did the universe start, and what is my thought, what is consciousness, right? And so that's why I'm working on the second, because frankly, I started working on the first, and that didn't quite work out when I was in graduate school. What do we expect to find when we study the brain as a physicist, right? And neuroscientists, biologists look for some things. We as physicists, at least I as a physicist, look for others. And here, another beautiful essay that many of you probably have read, What is Life by Schrödinger. And there he talks about that living matter, while not eluding the laws of physics that we know of exist to date, is likely to involve other laws of physics, which we haven't yet discovered. And when we discover them, they're going to be as part of our undergraduate curricula as the laws that for the inanimate matter, which we know, right? And so that's what I'm interested in as a physicist. I'm interested in trying to understand general laws. And the laws to me are interpretable, simple patterns which are simple and explain a lot of things, where with a lot, very little in, you get a lot of out, right? So that's what I call a law rather than a particular model. And we'll see whether we have reached this, at least in some small corner somewhere of biology. So why do we need new laws, right? The old laws actually work quite well. So this equation that you can see if you are sitting close by, this is just a simple, what would have been an introductory ENM. This is just our RC circuit equations, except that these are taken from a Nobel Prize winning work by Hodgkin and Huxley on the conductivity, conductance of a squid giant axon, right? So underneath all of biology, there is physics, right? There are molecules, they are really same molecules, whether we're talking about a piece of metal or whether we're talking about a squid giant axon. There is no life force, right? The only forces we know are the forces that are part of the standard model plus gravity, right? And so old laws work quite well, except that they get overwhelmed. So this is a reconstruction of ENM reconstruction of some neural network in some part of the cortex. This is a network of genes interacting with each other around a single gene MIC, which is what we were working on when I was in Columbia, as Anton pointed out. And so for every one of these different corners here, there would be laws that look like this, which are talking about the motion of various currents, or the motion, is there electric currents or probability currents or material currents, whatever it is, from one node in this network to another. And at some point you realize that there are more parameters in these models than there are space in your computer or in your notebook. And a friend of mine in Vanderbilt, he actually coined a new measurement, a new unit in science, which is called a Leibniz. And a Leibniz is an Avogadro number of differential equations. And so that's what you very quickly get to if you start applying the old laws, quote unquote, to systems, to biological systems one by one by one. I don't think it's a practical way of trying to model a human by building a human on a molecular scale somewhere on a computer. So where we're sitting right now in biology is that we have generated a lot of data. We have these big models, and we're approaching what Borges maybe warned us about, which we shouldn't do, which is trying to model an empire with a map that coincides with that empire on a scale of 1 to 1. And sort of the only use for that map is then to cover yourself from the rain and sun, just like in this illustration, because map is supposed to be smaller and simpler than the original system that you are trying to model. And in fact, in biology, this is what we have. This is just different various atlases. This is gene expressions in the brain done in the Allen Institute, where for every cell inside the brain of a mouse, we're measuring expression of every particular gene in that cell. Or there could be different sequences of some organism. I don't even remember which ones are this, how they relate to each other. Again, obscene amounts of data trying to be put together in big databases, right? And it's not just me or other physicists who say that this is maybe not the only way of going forward, of just collecting data and cataloging it somewhere, but it's even people who started the molecular biology revolution, like Sid Brenner, who tell us that biological research is in crisis. We're drowning in the sea of data, searching for some theoretical framework with which to understand it, right? So where would this new theoretical frameworks and new laws would come from in this very large dimensional biological systems? And of course, where would this new laws that I'm talking about, sort of new framework, come from? And again, we're not being original. I'm a statistical physicist. We're looking at systems with an Avogadro number of components. Where they should come from is from the same old, more is different. We need to be looking for statistical mechanics and try to find some kind of universalities that are not describing a particular system, but maybe a larger class of systems. One can always ask, is it necessarily the case that biological systems would have a low-dimensional emergent description, right? It's not a given. The fact that we have been able to use physics to explain metals doesn't mean that we would be able to use physics to explain the brain, right? So the thing is that there is a pretty good reason to believe that low-dimensional simple descriptions should exist in biological systems, and the simple reason for that is that we exist. We'll learn how to walk. We'll learn how to talk. If I had to set up the values of synapses in all of my 10 to the 11 neurons inside my brain, and I had to set them exactly right, if every single thing mattered, there is no way from just 1 year worth of experience I would be able to learn to walk, right? It means the fact that biological system can learn itself means that it is functionally a lot simpler than what the structure of it suggests. And so maybe we should also be that lucky, and maybe we should be able to at least look for and will be able to find those simple descriptions that we find in ourselves when we're learning ourselves. And so the next question that one can probably ask is why finding this more is different, why finding this different in living systems is harder, why we haven't been able yet to do so, even though a lot of smart people have tried. And the reason is that all the traditional physics tools for finding large-scale descriptions that we have developed in the second part of the 20th century, so most of them go to what's known as the normalization group. They are based on the ideas of symmetry, locality, homogeneity, no internal states for various components for the system, and no function. Systems are typical rather than existing in very special points in space. And all of that thing breaks down in biology. Brain is not symmetric. Connections are not local. You can throw an axon far away out, right? And brain doesn't operate in a typical configuration. It actually does something very, very specific. It was adapted and evolved to be in a very atypical state, right? So all the tools that we have developed, we don't yet know how to apply them for systems like that. And what's worse is our intuition fails. I'll show you a bunch of examples, things that we believe should be interesting. Maybe in fact they're not that interesting. Maybe they're essentially given to us, right? That's some not interesting part of biology, but it's just the statistics that shows itself up. And these things that are really interesting, maybe we're not even measuring them or studying them. Again, I'll show you the examples. So we don't know what is surprising, and therefore we don't know what we need to explain and what we already know how to explain, right? And so to move forward for the rest of the talk, what I'll try to build on are some of the ideas for building sort of theories, models of different corners in large dimensional biological systems. And the ideas will come from the field of physics, which is known as random matrix theory, random networks, where you're putting together a large number of interacting components with very specific structural rules. And it turns out, and this rules, this constraints maybe reflecting biology, and it turns out that the activity, the behavior of such system may in fact be universal and may not depend on the specifics of what you are trying to put together, in the same spirit as not everything matters when we're trying to go from micro to macroscopic scales. And so what I will also show is that some of these random models that we put together to explain biological data sets are equivalent, mathematically equivalent to a system actually exhibiting and exhibiting a low dimensional latent descriptions, effectively mean field-like descriptions that one can hope later to use to describe the dynamics of the system, even though we haven't quite reached that state yet. So before going any further, maybe what I will try to point out, just not everybody even in physics departments knows what random matrix theory is, which is maybe a shame. It's a part of theoretical physics of the 20th century, which I think is one of the more important ones and one of the most underappreciated. So I'm just going to give you one example. It's not the original example from Wigner's model of nuclei of heavy atoms, but maybe it's a bit more relevant to those of you who work with data nowadays. Okay, so suppose you are measuring things. Suppose you are recording 1,000 neurons from the brain, their activity, yes or no, are they spiking or are they not spiking at any particular moment in time? And so I have my 100 variables, 1,000 variables, and I measure them 1,000 times, right? So there are, the dark dot would be, yes, the neuron decided to spike there, and the orange thing would be that it didn't spike at that particular point. And so the first thing that any scientist who is going to do looking at this particular data set, you are going to start to analyze who is correlated with whom. Let's calculate covariance matrix of all the things that we have measured, right? That's what we do in condensed matter physics. We calculate covariance matrices between different points in the material and things like that. So let's calculate covariance matrix of the variables in this type of a data set. Let's just do it sort of step by step. Let's suppose I'm going to start with just 10 variables, and I'm going to measure them 30 times, right? So 100 measured 1,000 times. I'm going to start with 10 and measure it 30 times. So I'm going to calculate the covariance matrix, 10 by 10 covariance matrix of these variables. I'm going to calculate its eigenvalues, and I'm going to do two different independent times. Variables are by constructions uncorrelated with each other. They're just independent zeros and ones that I threw in this type of matrix. And so you will get your 10 eigenvalues, and there is not a specific pattern that you could see. You go to something larger. Maybe I'm going to measure 100 variables 300 times, calculate 100 by 100 covariance matrix, get a spectrum, and now you see that there is maybe some kind of a structure starting to emerge. By the time I get to 1,000 variables measured 3,000 times, which is more or less the numbers that I'm going to be showing you later today from biological experiments, and I calculate the covariances between the two. First of all, you see that they are essentially repeatable from one to another, right? And also, even though you started with data which in the limit of infinite sample will have no correlations, in reality, from finite sampling, you have the spurious correlations which have a very specific structure, which is known as the Marchenko-Pastur law. And so this is effectively central limit theorem, but the central limit theorem applied to matrices rather than applied to individual numbers. And matrices may have different statistical properties. So these ones are matrices of correlation structure. And for them, the eigenvalues will have this particular shape. For different types of matrices, eigenvalues may look differently. And that's sort of this whole big field of random matrix theories trying to calculate properties of large matrices with different types of constraints, realizing that maybe asymptotically when they get large, you start getting some kind of central limit-like results, right? And so we're going to try to use some of these ideas later in this talk. Okay, so now let's go to real data. This is actually a recording from biological experiment. Biology certainly has more, right? Anderson's more is different. We certainly have more. What I'm showing here is about 1,000 neurons in the hippocampus of a mouse, recorded for couple of minutes, and so there's some number of, and every time when you observe a spike from every particular neuron, you put a black dot there, right? So it's 1,000 neurons recorded for a certain amount of time. And this is what a typical modern biological data set is going to look like, except that it could also have been a different biological data set, right? So for example, I could have had a single neuron, which I record for a certain amount of time. That could be a neuron where a fly is staring at an outside world, and you are recording from the neuron what it is that it's seeing, right? The neuron is spiking or not, depending on what the fly is seeing in the external world, and you're recording it, and then you're going to show the same movie to that fly again and again and again and again. And so that's a repetition number, and you will see that sometimes you get a spike at the same time, right? Same object moving in the visual scene creates a spike, and sometimes it doesn't, right? What you would have, if you were working in a different part of biology, the same data could be the species number and then position in the sequence, and the dot would be a mutation. So think about it where this is the COVID genome, and these are sequences of viruses from different individuals. And every time when they're different from each other, we're going to put a black dot, right? This is, again, a typical biological data set. Or if you're working on developmental biology, you would have 20,000 genes. And these genes could be either active or not active, expressed or not expressed, open for transcription, not open for transcription. And then there would be maybe cells, different phenotypes, different types of cells that you are measuring from during development. And again, the data would come as this big matrix of either 0 or 1s or real valued numbers between 0 and some big number. Okay. So are there any interesting laws that, or at least maybe we don't yet understand what they are, but some interesting observations that people have seen in this type of data over the last few decades? And it turns out that there are actually very similarities between all of these different fields as soon as you start looking at them, not as representing a particular experimental system, but just a big bag of data, right? A matrix of data. So let's walk through these different plots. So over here, what I'm plotting is, this is the immune repertoire of a fish. So now everybody after a couple of years of COVID knows what antibodies are, right? So you take all antibodies in the fish, you sequence them, you identify if they are similar or not to each other, and then you plot them in the following ways. If the most common, the second most common, the third most common, the fourth most common, and so on and so forth, this is called the rank. And then on this axis, you put the frequency or the probability. How often does this antibody show up? Okay. And so what you will see is that for many different fishes, there are like 10 of them here, they all have this 1 over rank dependence of the probability of seeing a particular antibody as a function of the rank. This is called the Zipf's law. It was first observed in English language by Zipf. The idea is that the nth most common word in English language shows up with a frequency of about 1 over n compared to the most common word, which is the article be, right? That's the most common one that you will see in English. Well, that's great. This is immunology. Let's look at the recordings, neural recordings. So this is now recordings from a salamander retina. So a salamander retina is taken out from the animal. It's put in front of a screen. Movies are shown to the salamander retina, and you observe zeros and ones when the retina spikes or not, right? Each particular neuron is spiking is either 0 or 1. 1 is spike, 0 is no spike. And you look at this very long sequence of zeros and ones, and you call it a word, right? There could be a word, nobody is spiking, 0, 0, 0, 0, 0, 0, 0. Or this particular neuron is spiking, so 0, 0, 1, 0, 0, 0, 0. That's a word, right? And so you take all of the words that you see in the recording, you again figure out what is the most common one, the second most common, the third most common. You look at the rank, you look at the frequency, you plot frequency versus rank. And here we have a control. We can look at, or as a parameter, we can look at short words. I'm going to look at 10 neurons or 20 neurons or 30 or 40. And the longer sequences you look, the closer you are to the 1 over rank word, right? The larger the system, the closer it is to the Zipf law. This is again a neural recording. This is from our own paper, where I look at that fly that I mentioned to you, which is staring at the outside world. Now I'm going to look at words which are not across many neurons, but from one specific neuron over time. Is it spiking 1 or not? 0. I'm going to look at that sequence of zeros and ones, count how many times I observe a particular word of length n, order rank versus frequency, and I get the Zipf law, but only for words which are long. 40 or 50 milliseconds, or 40 or 50 zeros and ones in the sequence, while shorter words have not 1 over f, but some kind of a weird other statistics. And look at HIV. This is the specific proteins inside the HIV sequences from within a single individual. And again, the HIV is famously known for being extremely diverse even within a single individual. There are multiple different versions of it. And you sequence all of them, and you plot rank versus frequency. And for multiple different proteins, you get something that's really, really close to 1 over f. And so you'll have to believe me that essentially any data set of the type that I have shown to you, be that from neural systems, from gene expression systems, from whatever, if you plot it this way, you almost always get the Zipf law under some conditions. And then the question becomes why? These are all coming from extremely different systems, and in every one of these corners of biology, people have made a lot of statements about how it is important for this type of system to have this critical scaling behavior because it allows better computation or better fitting to the better working immune system, et cetera, et cetera. But it cannot be that the same law shows up in all of these different corners for all the different reasons, right? Maybe there is a simple explanation. Maybe it's not a statement about biology, but it's a statement about the underlying physics of the data that we're dealing with. And that's what I'll try to point out how this would happen. So first, let's talk about why Zipf law is interesting, right? So there is an interesting paper produced by Bialek and Moore a couple of years ago, where what they've shown is that if your rank versus frequency plot is Zipfian, sort of has a structure of 1 over frequency, has a structure of 1 over rank to some power. And I will look at that probability distribution, and I will call this probability distribution over sequences. I will represent it as log of that as an energy. So that starts looking like a statistical physics system. And I will count the number of sequences which have the same probability, that is the same energy, right? I'll take a log of that. That's going to be called the entropy. Then as soon as I have the Zipf law 1 over R, then the energy and the entropy in the system are proportional to each other. Okay, but if the energy, again, which is the log probability, and the entropy, which is a log of the number of states which have the same probability, are proportional to each other, then by the definition of the heat capacity, which is the inverse of the second derivative of entropy over energy, you will see the heat capacity explodes, right? The second derivative of S with E is 0, which means that if we were to treat this system as a statistical physics system, heat capacity is infinite. It's a system at a critical point. We should be all excited, right? That's sort of the idea. And that's where all of the arguments about why Zipf laws in different corners of biology are interesting, they come from. They come from this idea that critical systems allow very large susceptibility to inputs. Heat capacity is large, right? Large susceptibility. And as a result of that, the system then behaves in the most responsive way to the external stimuli, right? And so that must be good, right? We're responding to the outside world. Biology is really working very hard to get to those points because, again, remember, in physics, we don't get critical behavior for nothing. We have to fine tune to them. Except that it turns out that normally in physical systems, we work in systems which we work very hard to keep closed. Biological systems are not closed. The same neurons are observing external stimuli, right? The movies that the fly is observing. The immune system is being bombarded by pathogens externally. We do not keep these systems closed. So what if we try to build a model of a bunch of simple components talking to each other, which are being subjected to an external driving? What will happen, right? So let's try to do this. Very simple algebra. I'll promise it's like the last algebra slides that I will have. I will have the probability of all the spins that I have, all the variables, zeros and ones are uncoupled from each other. It's a really boring system, right? It's a magnet where every neuron is just, every spin is independent of everybody else, except that there is a magnetic field, or maybe we'll later talk about 5 or 10, some number of different fields, which are all coupled randomly with different coupling constants to this neurons, right? So what I have is total probability of all of the spins is a product of probabilities of individual spins conditioned on the value of the field. And then the overall probability, I need to integrate the conditional probability of the spins conditioned on the field over the values of this field, right? So I have the external field and I average over it. Turns out it's not a very hard statistical physics, large deviation theory calculation to show that in this system, as long as the spins are susceptible, right, susceptibility is not 0 to the external field, as long as these J's are random, doesn't really matter how random, as long as basically they're not fixed, right? There is some variance there. And as long as n, the number of spins is large, you're going to get a Zipf law. And the argument is actually very simple. We can follow it in a few lines. So what I wrote down before is probability of spins given the magnetic field was that they're all, it's an independent product of the spins. But in a statistical physics system, if the number of spins is very, very large, then the magnetic field corresponds essentially one-to-one to a particular magnetization. Right? It's going to be order square root n fluctuations, and that's about it. Right? It's almost the same as P of sigma conditioned on the magnetic field. But then all spin configurations with the same magnetization have the same probability. And so it means that this probability is 1 over the how many spins there are that corresponds to a certain magnetic field. That's my omega factor. And that is, by definition, e to the minus entropy of the system at this particular m. Okay. Now, if probability of the field is of order 1, meaning it doesn't change as I add more and more and more fields, more and more particles into the system, then what it will mean is that the overall probability of sigma averaged over the h is also going to be proportional to x to the minus entropy. That's it. But by definition, probability of a sequence was e to the minus energy. Right? By definition, energy was a log of the probability, which means that entropy is equal to energy in the system, which, as I explained to you before, would mean that we're going to get the Zipf law. And you can make this argument a lot more accurate. You can show that it gets even better when you have more than one field, up to order 1 fields. It must not be scaling together with n and so on, but you cannot escape it. As long as you have an external driver, a latent variable, effectively a magnetic field or mean field in the system, which is coupled to otherwise decoupled or weakly coupled units, you're going to get the Zipf law, which means all of the things that I've shown you before do not require any special biology to have, right? The only thing that they require is that there is an external driver, a stimulus for the neurons, or changing pathogens for the immune system, or whatever else for a particular system we're dealing with, and you're guaranteed to observe the Zipf law. And that's maybe an example of, I would say, a new law, right? It could have been discovered in physical systems, in inanimate physical systems, that if you drive the inanimate physical systems with magnetic fields, you will observe this very interesting weird statistical properties, but it wasn't, right? So that's a law that we discovered once we started dealing with biological data. There is an upcoming paper together with David Schwab and his postdoc, where we show that the existence of the Zipf law is essentially one-to-one corresponds to latent factors in these physical models, right? That if you have latent fields, you will get Zipf law with a few conditions. And if you see Zipf law, it's the same as having effectively a latent factors, which could either be externally imposed or emergent mean field-like descriptions. So this is an example of what I said before, that for this type of systems, our intuition fails. A lot of people have been arguing that this type of scaling behavior is interesting biologically. And my argument is that they are not interesting biologically. This is part that you will see in any biological system. What makes a system, what really stands for biology are the deviations from these universal laws, right? What biologists should be measuring is not the statistics which produces this laws, but they should be measuring things which allow us to say how different we are from the Zipf law and which places are away from the Zipf law. And unfortunately, such experiments are usually not done, right? Because people don't understand that this is what needs to be done. So, but let's try to go more with this. So a good theory needs to explain more than just one thing, right? So this latent variable coupled to non-interacting components, non-interacting spins, so far explains the Zipf law. Can it explain more? So we're going to talk very briefly about two more corners. One of them is temporal and spatial correlations within large neural recordings, and the other one is what's called neural avalanches. So let's look at this experiment. This was done by David Pang's lab at Princeton. This is a mouse which is on a virtual track, right? So it runs in a place where it's immobilized. We're recording from its brain. This is hippocampus of this mouse. about 1,000 neurons there. We're measuring activity of every one of these neurons, and it's immobilized on a ball. It runs on a ball, ball turns under it. This way you know which way the mouse is running, and you can put it in a virtual reality. So that if it was running on the ball left, then the whole picture in front of you turns left, right? And so the mouse is running on this virtual in this virtual thing, and you're recording this huge activity patterns, just like I showed you before. The question is, what do you do with it next? And so there's been a group of people led by Meshulam, who is currently in Washington, University of Washington, where they tried to figure out, can we detect emergent large-scale degrees of freedom in these hippocampal recordings? So the only thing which we know how to do is coarse graining, basically Kadanoff style block spinning, except that we cannot really do combining spins which are close to each other into a hyperspin. Why? Because there is no locality. Two neurons which are next to each other in this recording do not necessarily talk to each other. And two neurons that are far away from each other might actually be very close to talking to each other, right? So maybe what they suggested we should do is to combine together units which are not geographically close, but units which are strongly correlated. So correlation is a measure of, in some sense, locality, right? You are mostly correlated to people who are next to you, right? And so that's what they did. You take this neural recording from each individual neuron, you find the most correlated ones, you combine them into a cluster. Now of two neurons, it's our hyperspin or a hyperneuron, and then you combine these clusters into a cluster of size 4, and all the way like that. Since we have about a couple thousand neurons in the recording, we can do it many times, right? 10 times, roughly speaking. And this is a coarse graining transformation. The next thing that you would try to do, you would try to understand how various correlation functions in these recordings depend on the scale of this transformation, right? What happens to them after you coarse grain by 1 step, by 2 steps, by 3 steps, and so on and so forth. That's what any statistical physicist would do in a system like this. Great. So what do we expect? We expect that there might be some kind of a scale for spatial and temporal decorrelation. Once you coarse grain beyond that, correlations disappear. Since clusters are sums of neurons, neurons are random variables. If they were completely independent, you would get the variance of the activity of a cluster to go linearly with the size of the cluster. Just the law of large numbers, you add up random things together, variances add. If neurons were exactly copies of each other, then when you add n things together, variance goes as n squared, right? So we expect something either, dependent would be n squared, independent would be n. And then what we would expect is that the distribution of cluster activities, right, should be Gaussian eventually, because I'm adding up a lot of things together, okay? This is what's been observed experimentally. You see that the activity variance as a function of the cluster size is neither quadratic nor linear, somewhere in the middle. Beautiful scaling, right? Over a couple of orders of magnitude. You don't often see things like that in a condensed matter system. Even, here we're talking about a living mouse. You can look at the scaling of eigenvalues in the activity of these clusters. Let's not go into details of what this really could mean. And it has some certain power law. You can look at the correlation time as a function of the cluster size. And there is a beautiful power law, right? So there is no cutoff after which the clusters are uncorrelated with each other. So what we do is our favorite latent model, a latent features model, where I have a bunch of neurons and I have a bunch of latent fields. Each one of them is undergoing random walk, Einstein-Uhlenbeck process specifically. It's a random coupling, set of random couplings between the two. Really doesn't matter which ones we choose. We have explored a lot of different ones. And what we're going to try to do is to repeat exactly the same thing as Meshulam did on the recordings, but we're now going to repeat it on this latent variables model. This is what we get. The same scalings, and I'm just giving the numbers here. Experimentally, they measured the scale of 1.4, we got 1.367. Over here, they measured scaling 0.71, we got 0.65. For temporal correlation, there is somewhat of a difference. We know why, right? It would be actually bad if there wasn't a difference. We can talk about it later, right? So essentially, all of the measurements that they've done on their mouse are completely recovered by a model with a bunch of latent variables going on standalone back dynamics coupled to otherwise uncorrelated neurons. Perfect. Let's move. Oh, yes, there's one more feature here. Remember, I told you that you expect distribution of cluster variance clusters to be Gaussian. This is what you observe experimentally. This is what you observe in our model. Same behavior, a peak, a shoulder on the right. It's more than just qualitative similarity between the two. Okay, now let's look at one more example. It's like just seems like I cannot stop. There's something that is called Avalanche criticality in neural systems, discovered in about 2003 by Beggs, who is now in Indiana University in Bloomington. The idea is the following. You're going to record from, in his case, neurons which are taken out from an animal and put in a dish. And you will see sort of things that look like this. Nobody is firing, and then a bunch of neurons are firing, a bunch of other neurons are firing, and eventually, again, nobody is firing. So this is going to be called an avalanche. An avalanche, you can measure how long it is, and you can measure how many neurons participated in it. And you can plot the distribution of the sizes of the avalanches and the distribution of durations of the avalanches once you recorded them in an experiment. This is what you get. This is distribution of the sizes of the avalanches. There is a clear power law. This power law is going to be measured by an exponent tau. This is the distribution of times of avalanches, going to be measured by the exponent alpha. And this is now we can plot the duration versus the size, right? So it's not the distribution, but the duration for a particular avalanche versus its size, and still a power law here, and that's going to be measured by an exponent called gamma. There is a theory in condensed matter physics of what's called crackling noise due to Jim Cessna's, where he has calculated that for some crackling, for some interesting non-equilibrium phenomena, the relation between the exponents should be like this. Gamma is alpha minus 1 divided by tau minus 1. And in all of these experiments, by now, there are literally hundreds of papers where people have measured avalanches in any corner of neural systems, in vivo, in vitro, recordings, calcium, LFP, doesn't matter, like all of any kind of recordings. You more often than not observe avalanches. The actual values of tau, alpha, and gamma might be different, but this relationship holds. Okay? And you would not be surprised at this point where I take as the same model with latent variables, about 5 of them. They are going unstandalone back process, random coupling to a bunch of neurons which are otherwise uncoupled. And I get the taus, beautiful scaling for the avalanche size, beautiful scaling for the duration, and then the scaling between the size and the duration gamma, and the ratio of alpha minus 1 divided by tau minus 1 would be 124, and experimental field fit gives you 124. So it's a perfect fit to the avalanche scaling. Even avalanche scaling can be explained by this type of a model. So basically, at this point, we can say that there is a kind of a new law that we could have observed earlier or could have seen earlier in inanimate systems, but we didn't. Many measures of criticality are explainable by models with a few order 1 latent variables. Our intuition clearly failed. We thought that this would be a biological phenomena, but it's simply a statistical physics phenomena. Biology is a deviation from that, right? But these critical scalings are just a simple physics of it. And the next question we can ask is, where do these latent features come from? Because, for example, if you're recording from neurons on a dish, there is no stimulus, right? They're just actively talking to each other in a way that they manage to connect to each other, but there is no stimulus. So where do these latent variables come from in a system like that? So I don't know, when should I end? In 5 minutes. I'll try to walk very quickly in the next, try to give you an explanation of where this comes. And that's another biological experiment. This one is from Anne Churchland's group, now in UCLA, with the first author Najafi. She is at Georgia Tech. So here the idea is that a mouse is trying to make a decision. Did I hear Poisson clicks with a frequency of 10, or did I hear Poisson clicks with a frequency of 20, right? And as the mouse is trying to do this, we are recording the activity of both in excitatory neurons, so neurons whose activity excites other neurons, and inhibitory neurons whose activity inhibits other neurons in the, I think it was inferior temporal cortex. Actually, I don't remember where. In order to explain the statistics of the excitatory and inhibitory neurons, they needed to build a very detailed models of how excitatory and inhibitory neurons have to talk to each other. And this is sort of the summary of this model, where for every arrow there is a differential equation. You talk to that guy, et cetera, et cetera, et cetera. And we can ask, do we really need that? Do we really need this detailed network? So what Audrey and I did, we just created a random network with random couplings, with 20% neurons being inhibitory, 80% being excitatory, which is roughly speaking what you see in this part of the cortex. They are balanced, meaning that in order to have the overall activity that neither doesn't go to infinity, doesn't go to zero, the overall amount of excitation inhibition has to be about the same. So it's a biological constraints on the random network dynamics. And after that, we're going to simulate this networks, and we are going to ask how accurately can this network distinguish whether the stimulus is 10 hertz or 20 hertz. And this is the type of data that we would get, where in different instances of this network, they are accurate between 70 and 85%, more or less, to be able to distinguish which way what stimulus was presented. And experimentally, this is exactly what was measured, between 70 and 85%. It gets to be even more interesting than this. Various weights, how you would put things, like you're going to be making a classification based on the activity, whether the animal is thinking 10 hertz or 20 hertz. So you're going to put some kind of weights associated with activities of different neurons, and the distribution of weights in ours and in their experiments and in our models is basically indistinguishable from each other as well. Again, everything that they have reported, this very simple random network model can explain. Okay, so why is this interesting? We're not talking about now the Zipf law, but it's just that the random network model has managed to make a computation. Should I go left or should I go right? Is it 10 hertz or is it 20 hertz? So when you look at the activity of this network, what you realize is that if I plot the eigenvalues of the different modes in the activity of this network, which would fall on a circle like this is your Wigner semicircle law, but for non-Hermitian matrices, right? And then you try to see what happens when you provide a certain stimulus. Some modes get amplified by the stimulus. So what happens is that your network has a bunch of collective activities that can be picked up by the stimulus, not very many, right? For a network of about 1,000 neurons, the number is about 20, right? And then the stimulus activates one of those 20 degrees of freedom that could be activated. And this was noticed analytically. We did basically simulational work, but analytically, same results were obtained by Larry Abbott and students working with him just a couple of months ago. So the point is that when you put in a network of random interactions together, the activity of this network is not all over the map. The activity of this network is low dimensional, right? Unless you screwed things up. The activity of this balanced network is low dimensional. And so it could be that this low dimensional emergent activities, of which just one is going to be picked up by your stimulus, are precisely those latent variables that are needed in order to explain all of these various scaling laws. So that's at least our hypothesis. And so what I've tried to do to tell you today is that we can actually see this new laws in biology, statistical physics style laws in biology. And it could be that these statistical physics laws are actually related to the structure, to the low dimensional structure of activities within the neural networks or molecular network that explain particular systems. And my dream is that this allows us potentially going forward sort of change the degrees of freedom. I should not, just like when I talk about a magnet, I stop talking about spins and I start talking about the magnetization, a collective variable. And that's a more useful macroscopic description. Then maybe with some of these ideas going forward, I can start thinking about neural dynamics, not as activity of 10 to the 11 neurons, but as an activity of a bunch, not one, of course, not magnetization. This is hopefully a bit more complicated than a magnet, right? But maybe thousands or tens of thousands of latent variables, latent features inside the system. And similar with population biology, we should not be tracking every mutation. We should be tracking some collective activities which will show up there if we just look for them. And so with this, I'm going to end. Thank you.